Okay, so they didn't have true teeth. They had, we call them horny teeth, right? Horny teeth just means hard teeth, and they're composed of a protein. We looked at two different groups here in these particular Nathans. We looked at the lamprey, right, which was filter feed, was a parasitic as an adult, right, and it was filter feeder as a larva. How long can the larval stages last? It was up to 7 years. Okay, so larval stages can last up to 7 years. And where did the larva live? Obviously, both adult and larva are aquatic, right? Okay, but what different types of aquatic environments do they live in?
Shallow.
The larva lives in freshwater, right? Whereas the adult lives in marine or saltwater, right? And so it is the change in the water, right, that migrates into that causes a sexual maturity, right, and the change in its feeding mechanisms as well. So initially it does live as a filter feeder, and then later on, as an adult, it feeds on blood, right? So it's the blood of fish, so it's a parasitic. So that means if the larva is in freshwater and the adult is in saltwater, that the adults have to do what? Migrate into freshwater together, males and females, right? And then to lay eggs. And so they do have external fertilization. One of the characteristics of external fertilization is what? It only happens in water, aquatic environments, right? Only aquatic organisms have external environment. I mean, you can't dump eggs on the soil and expect sperm to swim across the soil, right, and reach the eggs or the ova, right? So it only happens in aquatic organisms, right? Either freshwater or saltwater. Okay, also, what are the chances that every sperm is going to swim to and fertilize every ova? Not good, right? Okay, so they produce large numbers of both sperm and ova to make sure that enough get fertilized that it's going to continue the species. Like, did y'all dissect the lamprey and lamb? Not yet. I know you have some there to dissect, so I'm sure you'll get to it eventually. But anyway, when you do, you'll see that it has a median gonad, whether it's male or female, right? Obviously, if it's a female, the median gonad is what? The ovary, right, that is producing the ova or the eggs, and they're all tiny little orange things that you can see about the size of a pin, right? And they're little orange things that are in the ova, and the ova is really folded and it's really long, so you can see that it's producing millions of little tiny eggs, right? And then the sperm, of course, are even smaller. So if you have a male, it's not going to have a large swollen gonad, right? The gonad is going to be flatter because sperm are much smaller, right? So you can kind of differentiate when you dissect whether you're dissecting a male or a female, right, by looking at that median gonad and the structure that it has. Okay, so they do have to migrate there to lay the eggs. They live as up to 7 years as filter feeders, migrate out, become mature, and start feeding on the blood of the fish. The other group we looked at there were the hagfish, right? And hagfish differed in their feeding methods in that they fed on what? Decomposing organisms, like when a dead whale dies and falls to the bottom and flesh starts to decompose. Decomposing flesh is pretty soft, right? Okay, so the lamprey basically just bore through it and get in gulps of food, right? They have teeth that are more elongated in their mouth, and so that helps to break off some of the decomposing flesh and funnels it back in to their mouth. And so if they feed on dead things, we call them detritus feeders, right? And so a lot of times those filter things out, but not this particular group. Okay, so they did have the tongue, and the tongue also has the teeth on it, right, that help facilitate feeding either on blood or other organisms. Did we look at other agnathans? Yeah, we did, right? Like the conodonts. One of the important evolutionary advancements of the conodonts was what? That they had teeth, right? They had a dentin teeth, right? So they had the dentin that secreted the calcium carbonate, right, which is very characteristic of a vertebrate tooth, right? But where was it located? What were they located on? It looked like spiral structures, right? Like maybe the tongue or something like that, all right? And so even though they had teeth then, did they have jaws? No, so they did have teeth. That was an important evolutionary advancement, but no jaws. What did they think they used the teeth for? Since they were circular, right, they could use them for cutting larger prey and taking off pieces of them, right, and taking them in. We do have some fish, the shark today called the cutter shark. Anybody familiar with it? It has like the rounded mouth, you know, with the teeth, and it basically, it's called a cookie cutter because it cuts out a piece of the fish it's feeding on, right, and leaves a scar there. So it was something similar to that, right? They had those rotating teeth, and so they could feed on objects that are larger than them, and they could break it up into smaller pieces, right? And then the other group that we looked at that were relatively small, that lacked jaws, were what? The ostracoderms, right? Ostracoderms did have some teeth, right? The ostracoderms were mainly what kinds? Where did they live? Were they free swimming, demersal near the bottom, or benthic? They were mainly benthic, they think, all right? They had sort of flattened bodies, and the chief characteristic of the ostracoderms was what? That their bodies were covered by bony shields, right? So basically, they had a bony type skeleton, endoskeleton that was just below the layer of the skin, and it helped to protect them. So the bony plates that covered the rest of most of the body were not fused together in one part, right? But rather made up of separate plates. And so the reason for that was what? Movement, right? If they were all fused, that would greatly limit their movement. And how do they have to move? By swinging their tail back and forth, right? Because they still have those myotomes attached to the notochord. They still have a notochord, right? So they still have those myotomes, the myomeres, the sections of muscles attached to the notochord. So they have to swing back and forth, right, in order for movement. If they had these bony plates all fused in one big plate, well, then what would happen? They wouldn't be able to move, right? They would be greatly limited. So the plates on the posterior parts of the body then allow for movement. They're in separate plates, whereas on the head region, the plates were fused, and therefore that gave them greater protection in that particular area, right? Okay, so these were relatively small, usually 3 to 10 centimeters. The next group, the placoderms, they also had some little bony plates on the surface of their skin as well, right? But they were much larger. Some of them were as big as 3 meters, which would be what? 9 or 10 feet, right? So they were much, much larger. And since they were much larger, is there probably not enough food for them on the bottom, right? So they were probably more free-swimming predators, much more like the sharks were, right? And so in both of those groups, though, what eventually happened to them? They became extinct. Did they leave any living ancestors or descendants? No, they left no living ancestors or descendants. So all we have that remains of them are fossil evidence, right, that they existed, and they did exist in large numbers up through the Silurian, and then even into the Permian. Permian's when we had the big mass extinction, right? And so that may be what ended that particular one. We're not really sure, but that's a possibility there. They think also the coming together of Laurasia and Gondwanaland, the plates moved those together to form one large continent, and it changed ocean currents as well, right, when that happened. And so that could have also contributed to their extinction. There's several things that could have led to the extinction of them, but anyway, we don't have them today, right? They're all extinct. Okay, so as far as the review goes here, then let's move on a little along here. So we have pointy teeth. Okay, now then the sensory pits. We talked about the sensory pits on the lamprey. They were located where? Around the mouth, right, and under the eye and above the eye. So they were located on basically the face area, right, the anterior region. And that's one of the things we saw in the evolution of animals, even in the invertebrates, right, is with cephalization. What was cephalization? The concentration of nerves and sensory structures in the anterior region, right? And we saw that it happened in animals with what kind of a body plan? Bilateral symmetry, right? And so here we sort of expect that, right? That there's going to be a concentration of those sensory structures, like the sensory pits on the anterior region. And the function of those sensory pits was what? Obviously, sensory pits are going to sense something, right? So what do they sense? They sense the vibrations and pressure waves, right? So like a swimming fish is going to do what? Create vibrations in the water, right? And so it can use that to find the swimming fish and attach to it, right? So that it can feed on the blood of the fish. So that's one of the things that helps it to find its prey. We looked at the eyes. Did it have very well developed eyes? No, right? So vision is not good. That's not really an important sense in finding its prey. With all of these particular Agnatha, not just the lamprey, but all the Agnatha, the conodonts, the ostracoderms, the placoderms, that their nostril was always all located where? In the middle of the top of the head, in the entire groups, all of those groups, right? It was, they had a single nostril in the top of the head. Obviously, the nostril is not used for respiration, but rather for what? As a sensory structure to detect chemicals in the water, right? So basically, it was an olfactory sense, and here it could smell the fish, right? The prey, and that was one of the chief things that led it to its prey. We know that that was one of the main senses, right? Because y'all haven't dissected yet, but we looked at it in class, some old dissection ones, right? And so we saw that right in front of the brain and right leading into the median nostril, there was a little black bulb, right, called the olfactory bulb. And so that was filled with sensory neurons, right? So you can see there's lots of sensory neurons that are detecting the olfactory sense or senses of smell. And then lying right behind it was the olfactory region of the brain. And the lamprey's brain was really tiny, right? But you could see that it was beginning to show the lobed condition, and that the olfactory lobes they weren't huge, and none of the brain was huge, but anyway, you can see they were an important region for it capturing its food. Okay, so we covered median nostril as well. On the external gill slits, okay, we talked about the origin of the external gill slits. So for the purpose of the exam, right? What was the origin of the gill slits? First, in organisms, we had what evolve? The muscular pharynx, right? The muscular pharynx was very important because it's muscular, so it could do what? Be a more efficient feeding mechanism, right? Instead of just bringing in water with cilia or something like that, right? A muscle's contractions are much more efficient in bringing in water for filter feeding, and then later for feeding on larger organisms, right? So that was an important adaptation for feeding. And then we saw that the pharynx pushed through the body wall all the way from internal to external, right? And that's what created the slits. And initially the slits were used for what? Filter feeding, right? Because the muscular pharynx could draw in water, right? And it could stick to mucus within the pharynx and then exit through the gill slits and then move it down into the esophagus and the mouth, right? And then later, ridges of vascular tissue developed on the inside along the top and bottom of the gill slits. And so that developed into what? The actual gill bars, the gill lamellae that contain the capillaries that could strain oxygen from water, take oxygen, right, and get rid of the carbon dioxide. So it became a respiratory structure. So the pharynx is very important there. in developing new feeding mechanisms, and that's one of the things we talked about is that if all the same animals are competing for the same food, there's not going to be enough, right? And so some of them are going to die off. But if some of them have a variation that allow them to feed on some other source that the others aren't feeding on, they're going to survive and pass that on to their offspring, so it gives them a greater chance of survival, and so it may result eventually in speciation or new species developing. Okay, we saw that they didn't have paired fins, they just have a single mid-dorsal fin. It was divided into an anterior and a posterior dorsal fin, and then they had a caudal fin. One of the characteristics of chordates is they have a what? Post-anal tail, right? So post-anal tail means it's where? After the anus, right? Okay, so they have a tail after the anus. Many of the invertebrates terminated at the anus, those that had them, right? And so that's one of the big differences there is the post-anal tail. And the post-anal tail, there was a caudal fin here, and it's called protocercal. Okay, protocercal means it's what? Partially circular. It circles around like this, and then sort of comes to a point. See one of my wonder? It's like here's the animal. It has a tail that looks, well, y'all saw it, right? When you looked at the specimen, did y'all notice? It's rounded here and then comes to a little point here. So that's, we differentiate it because it has a different tail. All of the agnathans have that kind of shape to their tail there. Okay, had no scales, just a leathery skin. Urogenital sinus and urogenital papilla. One of the characteristics we see in all of these gnathans, and later we're going to see even in the gnathostomes, is that they do have a common opening through which the gametes or sperm exit, or even the young, if they give birth to a lot of young, through which the solid waste exits and the urine exits, right? So that common opening is called the cloaca. I don't know if you like I raise birds. That's why I was a little bit late today. I have babies that I have to hand feed and took a little longer than usual. But anyway, a bird, when they defecate, anybody ever see it? There's like the solid feces that usually in the center, and then there's a liquidy stuff around the edges. So they're getting rid of both their feces, the solid waste, right, and the urine, the liquid waste, through that common duct, and they do it together. Not all, well, fish don't particularly do that. They can get rid of the feces, and then they can get rid of the urine, and then they can get rid of the eggs from at different times, right? But they use that common opening, the cloaca, for all of those. And one of the things that we saw is that it did have a little tube extending down called the urogenital sinus. So if it's urogenital, that means what? That what's going to come out? The urine and the sex cells, the gametes, right? If it's a male, the sperm. If it's a female, the ovum. Okay. Then, of course, the other important chordate characteristics we looked at, it does have that dorsal hollow nerve cord or spinal cord, and we saw that. So where is the dorsal hollow nerve cord located? Above the notochord, right? So it's dorsal to the notochord and extends the length of the body. It had the notochord, of course. Okay, this was on one of the quizzes already, but what was the notochord composed of? What kind of cells? Vacuolar cells, right? And so the vacuolar cells had vacuoles, which are spaces created by membranes, right? And what was inside the vacuole? Water, right? So that the water pressure within the vacuoles is what helps to maintain the shape of the notochord, right? And so therefore it is still employing that idea of a hydrostatic skeleton, right? Okay, we looked at the brain. Was the brain lobed? Yes, okay, it was tiny, but we could see the beginning of the lobes. So we saw the olfactory sac, gill pouches. The gill lamellae were the ones that grew up from the base and the top, right of in front of the gill slits there, and eventually formed respiratory structures. Okay, on the heart, we looked at the heart, right? How many chambers did it have in the heart? Two, right? what were the two chambers in the heart? The atrium, and what's the function of the atrium? It collects the blood returning to the heart, right? And then the other one was a ventricle, right, which does what? Pumps the blood out of the heart to all parts of the body. So which one is more muscular? The ventricle, right, because it's pumping the blood a greater distance. Basically, the atrium is just collecting it, right, and then contracting, forcing it into the ventricle right below it, right, and then it's forced out the ventricle through the ventral aorta, and then in the ventral aorta, we talked about that it branches, right, and so scientific terminology for the branches is afferent brachial arteries, but if we break it down into regular English, it just means what? Afferent means towards, they're going towards the gills, right? And brachial just means what? Branches, so it's just branches going towards the gills is what efferent brachial arteries means, right? So then they go into the gills, they break up into the little capillaries where they absorb oxygen, get rid of carbon dioxide, and then they come out the other side. So there they're called efferent brachial arteries, right? So that just means the branches that are carrying it away from the gills, right? And those eventually merge then into the dorsal aorta, which is going to branch off and to carry oxygen to all the major organs, right? Like hepatic artery, gastric artery, duodenal artery, right? So it's going to branch off into all those arteries that are delivering blood to the cells of the different organs and tissues there. And then once the oxygen is removed and it has waste material, it's going to go back to the heart and be recirculated again. So it has a two-chambered heart. Do all fish have a two-chambered heart? Yeah, all fish have a two-chambered heart, okay? And so they have just one circuit, right? It just goes out the heart, through the gills, to all parts of the body, and back to the heart. So it's we say it has a single circuit. Intestines, we know the intestines is for the food. Median gonad, meloprima, we did that. Okay, we did talk about the evolution of the kidney, right? And so the kidney is important, particularly whether they're living in freshwater or saltwater, and this does both, right? So in freshwater, it's going to take in more water, and it needs an efficient kidney to get rid of that excess water. So it really needs a more efficient kidney in the larval form, right, than in the adult form. But it does have two paired kidneys going down each side of the body there. When y'all do the dissection, you can look, right, there's like a little long flap going down each side, and they meet at the urinary papilla. And so you can see when you do the dissection of the lamprey, you can see those. And then, of course, an important characteristic showing segmentation is what? Remember, segmentation is one of the characteristics, right? Okay, so what shows the segmentation? The sections of muscles, right, that are attached to the notochord, so that when different ones contract, that produces the coordinated movement, right, of they're contracting, gets the notochord, that produces a coordinated movement, right? Okay, and so what were those sections of muscles called? Myomeres, all right? And myo means muscle, and meres means parts, right? So they're called myomeres, and so that's a very important characteristic there on this particular group. All right, so let's just review the sharks a little bit here real quick, and then we'll go on to the Osteichthyes. Yeah, these are some of the questions. Okay, so these are some of the parts of the shark, and y'all are dissecting it, right? So you probably already looked at some of those, so you can just tell me about them. Okay, what's the rostrum also called? The nose, right? And that's one of the things that we saw that with the shark, the mouth is located where? Ventrally, right? And usually the food is where? Usually it comes up on the food, right? And so the rostrum has to come back, right? And then the jaws actually come out of the mouth to capture the prey and pull it back in. And so it kind of changes the whole shape of the face, right? When a predatory shark is feeding. I'm sure you've seen it in nature shows like that. Okay, the external nares. Okay, here, unlike the lamprey, these are what? These are paired, right? And we did see sort of in the placoderms, that in the placoderms, the nostrils had begun to move from the central median nostril to move to a paired nostril slightly down, right? So we did, even though they left no descendants, right? We did see that movement in that particular group as well. And then the function of the nares, of course, is what? They lead into the olfactory bulbs, right? There are two of them, one for each nostril, filled with sensory neurons, right? That detect the olfactory senses. And so we can see that the olfactory sense is very important. We talked about if we wanted to film sharks, how would we do it? Chum the water, right? Throw blood and fish parts, and then it's going to disperse out, and the fish can smell small bits of the blood and fish parts in the water, and it follows a scent trail back to where it came from, right? So that it can feed on whatever was dying and pieces were coming off of it or whatever. Okay, so that's one of the first things that lead predatory sharks to their food, right? Is that olfactory sense or sense of smell. Okay, the mouth then we said is located where? Ventrally, right? And does it have two jaws? Yes, right, of course. What do scientists think the jaws evolved from? First pair of gill bars, right? And then the second pair of gill bars became the what? Supporting structures of the jaw, right? So the first pair became the actual jaws themselves. We looked at the gill bars on the shark. Did y'all do that? And I know the ones we had in here, we looked at it, right? And the gill bars are shaped like this, right? With the gill lamellae coming out here, and it has little gill rakers, right? Or gill teeth coming out the front of it. And so it has a similar shape of the jaw, so if it were to become calcified, right, we could see that it could function as a jaw. And so that's how scientists think that the jaws evolved. What are the teeth composed of? Okay, they're composed of calcium carbonate, right? They do have a dentin and a pulp, right? And all of that's characteristic then of a vertebrate tooth. So they are considered true teeth. Okay, what do scientists hypothesize the teeth may have evolved from? Okay, like the placoderms had the bony plates in the skin, right? Okay, and the bony plates in the skin and the shark are also present, and they're sort of like little teeth-like structures, right? And so if you look at it, it looks like little teeth, and so they're called placoid scales or denticles. Denticles means little teeth, and so the scientists hypothesized that the teeth in the jaw area, right, may have enlarged and formed the teeth of the shark. Anyway, that's one theory there. Do sharks, can you identify the species of the shark from the shape of the tooth? Yeah, you can, because we talked about they developed specialized feeding methods so that they have a better chance of survival. So if they're feeding differently, well then their teeth are going to be shaped differently as well, right? And so, yes, you can identify the species of the shark from the shape of the tooth. Do sharks periodically shed their teeth? Yes, right? They have rows of teeth that they're producing. Some sharks shed their teeth as much as every week, right? So that they always have fresh teeth available. And so those teeth can reach the bottom and wash up on shore and stuff like that. On the ampulla lorenzini, what was the function of the ampulla lorenzini? They're electroreceptors, right, that detect low-level electrical fields. Which sharks probably use it most efficiently? the hammerheads, right? Hammerheads basically can feed on animals that are on the bottom, like a flounder, right? It's buried in the sand, camouflage. It can, by swinging its head back and forth, it identifies the electrical field of the flounder and then goes down and eats it, right? So even though it can't be seen with the eyes, right, it can be detected because of that electrical field that it's giving off. Okay, on the eye, did it have good vision?
Yes.
Does it have color vision?
Yes.
What color is a tractor shark?
Red.
Reds and oranges, right? How do we know it has color vision?
That's cones and rods.
Because it has cones, right? The rods are for only black and white vision, so we analyze the retina of the shark, right? We find it has both rods and cones, so we know that it does have color vision, right? And then also we can test its response. Remember I talked about throwing the dummies in with the orange vests on and the sharks chomped down on them, right? So it did attract the sharks there. What was the conjunctivus? What's the function of it? To protect the eye, right? It's a little white lid, and when the shark bites into prey, it rolls up over the eye because the prey might have teeth or nails or something like that, or even a flopping fish could hit something in the eye, right, and blind them. So anyway, it protects the eye at the last minute when they're biting into the prey because a blinded shark would have difficulty feeding, right? Okay, the iris, the parts of the eye are like our own. So the iris is what on us? It's the colored part of the eye, right? And basically, what does it do? It regulates the size of the pupil, and the pupil does what? Lets the light into the eye, right? So like if you go into a theater, you know, your iris has a pupil pretty narrow, right? Letting in less light so you don't see for a while. And then the iris lets in more light right through the pupil, so eventually you see. You go back outside, you're blinded by the light, right? It takes time for adjust. So the shark has that same thing. It has the iris that can adjust for the amount of light. Obviously, if it's cruising along the bottom of the ocean, it's going to need what? Probably the maximum amount of light, right? Going into striking the retina. Whereas if it's cruising closer to the surface, it's going to have less light, right? Okay, so that the iris regulates the light in the same way that it does in our eye, and the pupil is the opening there that allows the light to pass in. It's not just a hole, right? Of course, it's covered by the sclera, clear sclera, right, which we call the cornea, right? So it does have that cornea as well. Y'all saw the lens, right? We took out the lens in here and looked at it, and what did the lens look like? Remember we took out the lens on the shark? Looked like a little marble, right? And it was opaque, right, rather than clear. So that shows that how little light, right, is necessary to stimulate the retina of the shark. How many gills did it have? External gill slits? It had 5, right? Except for there's one shark that has 6, and it's called the 6-gill shark. But all others have 5. And they have no protective covering like an operculum. Did they have pectoral fins? Yes, right. What's the function of the pectoral fins? Okay, basically the direction was one, right? And then also we talked about the shark's body is designed, and we could describe it as being what? It's torpedo shaped, so we could describe it as what? Hydrodynamically designed, right? Because the rostrum or the nose is pointed, so it breaks the water, right? And the water spreads out along the sides of the body. So that reduces the friction of swimming, right? Because of its body shape. So it means it has to use less energy to overcome friction in order to swim. And water is much more dense than air, right? So it creates more friction. There would be more friction to overcome in the swimming motion there. Okay, also they can provide lift because with the shark's body is heavier than water, right? So if it stops swimming, what happens? Sinks to the bottom, right? Does it die? No, some like the basking sharks, right? Basking sharks are filter feeders. They're the second largest of all the sharks. So they feed on plankton. When it gets really, really cold, is there as much plankton? No, right? So basically they just rest on the bottom until, you know, it warms up a little bit, and then they go feed on the plankton. And that's the second largest of all sharks, right? They don't die. They just sort of hibernate on the bottom of the ocean until more food is available. So that was a myth that, you know, the sharks died if they stopped swimming there. Okay, so those help to provide lift too. The pectoral fins and the pelvic fins also provide a little bit of lift that counteract the heterocircle tail, right, that swings back and forth. That swinging back and forth is called what? Anybody know the way it swims? It's called sculling, all right, and it's due to the contraction of the myomeres, right, against the cartilaginous vertebra. So here in these, they do have vertebra composed of cartilage that has replaced the notochord in the embryo. So the notochord is present only in the embryo, not in the adult, and it has cartilaginous vertebra that replace it. And remember, we looked at it in class, right, where the tail was cut. We looked and we saw the vertebra, and so we saw the circular area there that's called the notochord passageway, and so that's where the notochord was in the embryo, and then the vertebra formed around and replaced the notochord, right? Okay. On the reproduction in sharks, and this was true also of the placoderms, they found some a female placoderm with an almost fully developed offspring inside of her body. So they did have, some of the placoderms did have live young. And so the sharks, some of them do give birth to live young, whereas others lay eggs, right? We talked about that previously. If they lay eggs, that's called what? Oviparous, right? Because what is an egg? An ovum, right? So it just means egg laying is all that oviparous means, right? Okay, the ones that give birth to live young, basically there are several mechanisms by which they can do it. One is that they can just produce young with a yolk, right? And there's a little umbilical cord that's attached to the yolk, so they get their nourishment from the yolk to develop. And so gestate, like we looked at the dogfish sharks, right? And in the dogfish sharks, they give birth to live young, right? So they're called oviviparous, and they have that yolk that they develop. So in those, gestation takes 2 years, right? So they carry the young for 2 years before they give birth to live young, right? So gestation periods may be very, very long. Others, we talked about the great white, right? The great white, where it's like the squalus doesn't usually get over 3 feet, and it can produce like 16 to 30 babies, right? Okay, so great white, you know, it can get 18, 20 feet. It only produces 1 young at a time, so that means that the young is like 3 feet, weighs 125 pounds when it's born. So obviously the gestation period there is very long, right, for it to grow that large, and the first one that's laid develops first, and it eats up its siblings, and then once those are gone, once it uses up its yolk, and then it eats its siblings, and then the female continues to produce yolk, right, that goes down and feed it, right, and it continues to grow inside of the uterus. So those are different methods that different sharks employ. We talked about some that laid eggs too. Anybody remember those? Some of the sharks that lay eggs? One was the bamboo shark. A lot of people keep those in aquariums for pets, right? The bamboo shark lays an egg, and there's like a little thin thread that comes out first, and it wraps the thread around coral or something like that. And then once it wraps the thread, it pulls, and that basically pulls the egg out, right? And then the thread also holds it in place while the little shark is developing. And they have a thin keratin-like covering, you know, it's kind of a horny covering, right? That is inside, so you can actually, if there's a light, you can see the little shark inside with its yolk developing, right? And you can actually watch the development of the shark. The horn shark is another one that reproduces in the same way. A little thread comes out, wraps it, and it pulls the eggs out. Okay, so those are the, just reviewing some of those. The next group we're going to look at are the I had all of this on the flash drive, but I think somebody took it from my office yesterday. Somebody shared my office said somebody came in and took something from my computer. I think it was my flash drive.
It's not that flash drive. It's not that blue one.
It was an orange one, yeah.
All orange.
Yeah. This is my backup here, but I'm not sure it's up to date. Okay, that's about the point we're gonna start on for the quiz, right? The conodonts, right? So we'll go do conodonts through osteichthyes for the quiz on Thursday, right?
Conodonts through what?
Through the osteichthyes. Okay, a couple of groups here. Some of the earliest of the bony fishes here were in the Teleostomi, and so it's a large group, and it also includes Canthodians. And Canthodians just means spiny forms, and so the lead fin had little spiny processes that were attached to a muscle, and then it had a thin membrane, right, that was stretched from those little spines. Okay, the vertebral column was incorporated and ossified in a series of neural and hemal arteries along the nodal cord. So here it does still have some of the nodal cord, and it's becoming more ossified. Okay, the body armor is much reduced from the ostracoderms. Has a dermal plate around the head, but no head shield. Okay, so we can see the bony spines here, some examples. Okay, so they were, include the groups here, the osteichthyes. So literally, chondrichthyes meant what? Chondro means cartilage, and ichthyes means fish, right? So it meant cartilaginous fishes, indicating they had a cartilaginous skeleton, right? And in the chondrichthyes, the only calcified parts were what? The jaws and teeth, right? That was it. Okay, so here in osteichthyes, one of the things we see is that we have a bony skeleton, right? And so we did see that in the previous groups, like ostracoderms and placoderms, they had no living descendants, right? Okay, but they did have some of the same characteristics that involved in them that involved here in the osteichthyes. So those bony plates obviously were made up of what? Calcium carbonate, right, that covered the body. So it formed a sort of plate-like bony skeleton. Here in the osteichthyes, there are a few that have scales called ganoid scales, and so the ganoid scale does have a central calcified area, and then it has another enamel-like material, which is called ganoid, right, or ganoin. And so that's sort of reminiscent a little bit of that particular group, because it does have that little bony portion on it, and then the rest is another scale. But they don't have the same bony plates that we see in those particular groups. So some of the ideas on how this could have happened, with these, the bone may have been obviously stored internally, right, near the surface, and that formed the bony plates and the ostracoderms and the placoderms. The idea on the other bony fishes is that they had a cartilaginous skeleton, much like that of the sharks and skates and rays, right, the chondrichthyes. And some of the some of the osteichthyes have actually sort of reverted back to that cartilaginous skeleton. There's a group called the dipnoi, which is the lungfishes, and lungfishes have a more cartilaginous skeleton, right, rather than a bony skeleton, even though they are classified as osteichthyes, right? So it's sort of reverted back to that cartilaginous skeleton. It was a little bit more efficient for their movement because of their body shape. Okay, anyway, one of the ideas here on how the bony skeleton evolved is that, you know, the osteichthyes had this cartilaginous skeleton, and that some of them, remember that if they can move into an area that's not being exploited by other organisms, then they may have more food, more places to live and stuff like that. So they think that some of these bony fishes migrated into freshwater because no other fish were living there, right? There was no competition for food, okay? But they had to make some adaptations, right? And so remember that the ability to do the job has to be there before the job appears, right? In other words, they have to have characteristics that are going to allow them to do that before they do it. It's not going to evolve while they're there, okay? So anyway, as they moved into the freshwater, one of the things that they needed is a calcium carbonate, right? Calcium carbonate is not just for the bony covering like in ostracoderms or placoderms, right? But calcium is also used for what? Calcium ions. Why do we need calcium ions? Okay, because we need it for nerve transmission, right? At the end of the nerves where we have the synapses, right? Okay, there's a neurotransmitter, right, that's going to have to go across, and there's little vesicles of calcium ions, right? And the calcium ions cause it to be drawn up, and then the neurotransmitter released, right? So it stimulates the next nerve. So we have to have the calcium ions for nerve transmission, right? So that we can communicate with the body. Also, nerves innervate the muscles and cause contractions, right? And it's calcium ions that cause the contractions of the muscles. They flood into it and cause the contraction. So that we have to have them for nerve transmission and for muscle contractions, as though without them we probably wouldn't be able to survive, right? So the idea here is that in freshwater sometimes there was more calcium than at other times, and so when there was excess calcium, then they would store it in their cartilaginous skeleton, and then when there was not very much calcium, they could draw it from the skeleton. So the skeleton was acting like a reserve of the calcium carbonate, right? And they could use the draw from it for the calcium ions that are necessary. And we can see it's still like that today, right? We have a couple of hormones that help to regulate that, calcitonin, right, and parathyroid hormone. Example, my brother-in-law had really bad kidney stones, right? And they found it was caused by calcium, right? So the urologist told him not to eat anything at all with any calcium in it. So for like 15 years, he didn't have any calcium whatsoever, which your body has to have calcium, right? Because you have to have the ions for transmission. So since he wasn't taking it in his diet, where did it come from? His skeleton, right? So his body started taking the calcium from his skeleton because he had absolutely none in his diet. And so his vertebrae started collapsing, and when the vertebrae collapsed, they pinched the nerves that were exiting between the parts of it, and so it was resulting into paralysis, right? So to reverse the paralysis, they had to take bone from his hip and put it between the vertebrae, right, to free the nerve so that it wouldn't be pinched and reverse the paralysis. So we can see even today, right, that it still serves as that reservoir for calcium. When we have too much calcium in the circulatory system, well, then we have hormones, right, that cause it. A parathyroid hormone, I believe, is one that stores it in the skeletal system, right? If we need it, then calcitonin removes it from the skeletal system. Right? So basically, it still functions in that same way as it did in those ancestral fish. And so that's how they believe that the bony skeleton evolved. First in the freshwater fish that used it as a reservoir for calcium. Also in the bony fishes, they would have to have a very efficient kidney. We talked about that previously, right? Any organisms that migrated into freshwater, water is going to come in. The bony fishes do have scales, and the scales are sort of like the tiles on a roof. They're overlapping scales, right? And they're mainly composed of keratin protein, but there are different kinds of scales, like ganoid scales that have a bony portion, and then an enamel-like material called ganoin. One of the groups we have here have those kind of scales. You may know what they are. Found in the Rosacus. The gar have the ganoin scales. Yeah. So anyway, so they do have the overlapping scales. That prevents the entry of water into the body if they're in freshwater, right? But the part that has to be exposed are the gills, right? So they take in large quantities of water through the gills when they're taking in oxygen, getting rid of carbon dioxide. So they have to have a very efficient kidney in order to get rid of that. So bony skeleton, that's one of the major evolutionary advancements here, right? Another one of the major evolutionary advancements then is the swim bladder or air bladder. Because when we looked at all the other fishes, their body was heavier than water, right? So if they stopped swimming, what happened? They would sink, right? Anybody remember one thing that the shark did have to give it a little buoyancy? The liver is full of vitamin A oil, right? And so that vitamin A oil it floats, right? So basically that gives it a little bit of buoyancy. And the liver was the largest organ in the shark, right? Made up about 25% of the body weight of the shark. Okay, and we do need vitamin A, right? But if you were to eat all of a shark's liver, I guess depending on the size of the shark, it could be toxic because vitamin A oil is not water soluble. You can't get rid of it with the urine, right? It's fat soluble. So if you took in too much, it would accumulate in the body and be damaging. But you do need it. Like I have macular degeneration, which macula is the mid part of the eye, the conjunctiva under the retina, and so if you don't get enough of the vitamin A and lutein, so those are some you want to be sure you get while you're young because you're going to need it when you get older. Anyway, little deposits build up under the retina, and so as the retina is lifted, it damages basically it's in the area where the cones are, right? So it affects your color vision. Like my wife sent me to get the paint for our house, knowing that my color vision is not what it should be. It's about 50%, and so the painters put the paint on, and she's like, "Oh, that's not the right color paint," So I said, "I'm not going to spend another bunch of money repainting it. We'll live with it." So, that was her. She should not have sent me. She knew better. Anyway, so we do need those, right? And the shark uses it, and then he also uses it for buoyancy or flotation. Okay, so the idea here is that when fish migrated into the freshwater, areas of freshwater are very shallow, right? Shallow water warms up faster than deep water, right? And so when they were in this shallow, warm water has less oxygen in it than colder waters because the oxygen, when it has more heat, leaves into the atmosphere, right? So with less oxygen, either they died, right, or they got oxygen from some other source. So some fish would come to the surface and gulp air, and so if they gulp air with their mouth, where's it going to go? Down their digestive tract, right? And so they already have, on the fish that y'all are going to dissect them, I guess, in the lab. I brought some, but we didn't get to it today. Anyway, you can see they do have little pouches, little finger-like pouches around the stomach now. And so if some of those pouches then became enlarged and had capillaries, they do have capillaries anyway for absorbing nutrients, right? And so instead of absorbing nutrients, they absorb oxygen, and so that evolved an accessory source of oxygen. And we still see that today. Anybody have a beta for a pet? If it, they keep them in little tiny jars, right? Even in the shops. So once they use up the oxygen in that jar, what do? In the water, once they use up the oxygen in the water in the jar, they come to the surface, right? And they gulp air. And so they're getting oxygen from the air, right? And so they have this highly vascularized swim bladder that is used as an accessory source of oxygen. And some of them migrated into waters where the water would dry up, right? If the water dries up, they can't get any oxygen from it, right? So they develop very efficient lungs, right? So they still have gills when there's water, but they have very efficient paired lungs, the lungfishes, right? That they can use when there is no water present. Okay, then secondarily, some of these fish went back into the ocean, and in the ocean there's plenty of oxygen, so they don't need it for that. But what did it give them in the ocean? It gave them buoyancy, right? Now then, if they stop swimming, they float, right? So it provided buoyancy for those that decided to migrate back. Okay, now this is a very important group because this group gave rise to the land tetrapods, right? Because at this point we see that all life is in the oceans, which makes sense because the earth is 71% covered by water, right? And so if life is going to evolve, it makes sense it's going to evolve in that area that is the greatest there. Okay, there's several groups of fish, like we looked at the teleostomi, right? That included the Acantha, which had the spines, right? So here we have the spines and the little fleshy things there as well. So some of them then we called the ray fin fishes, simplified, right? And these are also called the teleos. The ray fin fishes have the little rays, and then they have the little membrane in between that they use for swimming that makes up their fins. And then we have another group called the lobe fin fishes. Okay, which are the most abundant, the teleos or the lobe fin fishes? The teleos are much more abundant, right? They're the most abundant of all vertebrates. There's so many different species of them, all right? But the lobe fin fish, there's few of them, but they're very important because they did what? They gave rise to the land vertebrates, right? The first tetrapods. And so there's several different groups here of the lobe fin fishes. So in the lobe fin fishes, rather than having the rays, they had what? Here's a sturgeon. We'll get there. Okay, here's one, the coelacanth, right? And so the coelacanth is a good example. The coelacanth was thought to be extinct, and in 1950 they started catching them off the coast of Africa, and then they caught another species off the coast of Indonesia. So it proves they're not extinct, right? And they've caught many, many more since then to study. Okay, these mainly use their fins for moving among, is it time? Yeah, it's time to go, isn't it? All right, moving among the rocks, right? So they have like bony appendages, right? Surrounded by muscle and flesh, and they use that for movement. And if we look at them, they have the same bones, right, in their fins that we have in our appendages, right? So they have a humerus, an ulna, a radius, carpals, metacarpals, phalanges. The only difference is on the phalanges, on each appendage, we see that there's, in the first groups of them, they had as many as 8 on each, 8 phalanges on each one, right? So that's 16 fingers, right? And then we see that another group had 7, another had 6, right? And then another had 5. So there was a tendency to lose those extra phalanges, right? Until eventually we ended up with the one with the 5 phalanges that gave rise to the land animals. Okay, so remember we're gonna have a quiz Thursday then from the Agnathans through the Osteichthyes. And then exam next week. Y'all have a good day.
What's the format of the exam?
It's gonna be, similar to